So, are you feeling totally overwhelmed by the sheer volume of biology you've got to master for NEET? If your backlogs are piling up, the syllabus feels like this massive mountain, and you're just struggling to keep up, listen, it's time to apply a really simple but incredibly effective mantra. Face it, fix it, and make it better. Welcome to This Explainer. Today, we're going to face those challenges head-on with the ultimate, structured, high-yield review of Cell the Unit of Life. We're breaking down exactly what you need to know directly from our source material into clear, logical steps. You don't have to be perfect on day one, but by focusing on these core concepts, you are going to get better and better. Okay, let's dive into this. Here's our roadmap for today. We'll be covering the foundations of cell theory, prokaryotes versus eukaryotes, the endomembrane system, semi-autonomous energy powerhouses, cilia and flagella, and finally, the nucleus and chromosomes. All right, let's kick things off with Section 1, Foundations of Cell Theory. Think about it, this whole journey kicked off way back in 1665 when Robert Hooke discovered cells by observing dead cork tissue. He was actually the guy who coined the term cell. Shortly after that, Anton von Leeuwenhoek made a massive leap. He became the very first person to observe and describe living cells. Fast forward quite a bit to 1838, and a botanist named Matthias Schleiden concluded that all plants are fundamentally made of cells. Literally the very next year, in 1839, a zoologist named Theodor Schwann hit the nail on the head and stated the exact same thing for animals. Together, they formulated that baseline cell theory, realizing that all living things are made of cells. But there was a pretty big missing piece to their puzzle. They couldn't actually explain where new cells came from. And that's exactly where Rudolf Virchow stepped in during the year 1855. So, the crucial takeaway here is the phrase omnis cellula acellula, which directly translates to all cells arise from pre-existing cells. Virchow added this brilliant piece of insight to finalize and expand the cell theory. And honestly, it's just mind-blowing that this single phrase completed the entire puzzle of early biology. Now pay close attention here, because this exact concept, knowing Virchow's name, the year 1855, and the meaning of this phrase was a direct PYQ, a previous year question in the 2019 NEET exam. It's an absolutely fundamental fact you have to lock into your memory. Moving right along to Section 2, Prokaryotes versus Eukaryotes. Now, I love this little trick to keep things straight. It's a simple mnemonic, P for prokaryotic, P for poor. Our source explains that prokaryotic cells, like bacteria, are fundamentally poor because they completely lack a true membrane-bound nucleus and they lack other membrane-bound organelles. They are very, very basic. On the other hand, eukaryotic cells, which make up plants, animals, you and me, are the biological luxury upgrades. They are fully loaded with a defined nucleus housing their DNA and all these complex internal structures wrapped in their own membranes. But before we get too deep inside these cells, we really have to talk about physical shape and size, because they vary wildly depending on what the cell actually does. The largest isolated single living cell is the ostrich egg, but the longest cell, that's the neuron, or nerve cell, found in our brains and spinal cords. It is absolutely vital to make this distinction between a large overall diameter and a long length. Examiners love to test the specific difference, so don't let it trip you up. Let's think back to our poor prokaryotes for a second. If they don't have those specialized membrane-bound power plants like mitochondria, how do they survive? How do they even breathe? Well, the answer lies in mesosomes. These are specialized infoldings of the plasma membrane in bacteria. Because bacteria lack those complex organelles, these mesosomes step up to handle all the essential survival tasks. They help with respiration, secretion, and they drastically increase the overall surface area of the membrane for vital chemical reactions. It's just an incredibly efficient biological workaround. That brings us to Section 3, the endomembrane system. Let's see how this all builds together. Inside a eukaryotic cell, you have all these various organelles, but they don't just work alone. The endoplasmic reticulum, the Golgi Apparatus, lysosomes, and vacuoles are uniquely grouped together into what we call the endomembrane system. Now, why just these four? Because their functions are highly coordinated. They don't operate in isolation at all. They rely entirely on each other to keep the cell's production and waste management running smoothly. Picture how this biological assembly line actually operates in a step-by-step flow. First up, proteins are synthesized by these tiny structures called ribosomes, which are stuck right onto the rough endoplasmic reticulum. Once they're made, these materials are sent on over to the receiving side of the Golgi apparatus. That's known as the cis phase. Inside the Golgi, the materials get modified, refined, and neatly packaged up. Finally, they get released as perfectly functional materials from the shipping side or the trans phase. It is just a perfectly coordinated pathway. All right, check out this actual trick question from the NEET 2023 exam. Which of the following is not considered a part of the endomembrane system? Based on that assembly line we just traced, you can confidently spot the outliers. Remember that independent organelles like mitochondria, chloroplasts, and peroxisomes are completely excluded from this coordinated system. Their functions are not directly tied to that endomembrane production pathway, which makes them prime targets for trick questions. Speaking of those independent guys, Section 4, Semi-Autonomous Energy Powerhouses. So we have these two main metabolic marvels operating completely outside of that endomembrane system, mitochondria and chloroplasts. Mitochondria are these double membrane structures responsible for aerobic respiration. They are the true powerhouses, just churning out energy in the form of ATP for both plant and animal cells. On the flip side, we have chloroplasts. Now, these are exclusive to plant cells and certain protists. They contain the green pigment chlorophyll to trap light energy, literally turning it into chemical energy through photosynthesis. Now, while chloroplasts give plants their vibrant color, plants also have these colorless plastids called leucoplasts, and this brilliantly illustrates their specific, crucial jobs. Because they lack pigment, they obviously don't do photosynthesis. Instead, they have very specific storage functions. Amyloplasts store carbohydrates like starch, think of a potato. Elaioplasts are responsible for storing oils and fats, and elaioplasts store proteins. Actually, memorizing this exact mapping is absolute gold for your exams. All right, Section 5, Cilia and Flagella. So how do these cells actually get around? Well, cilia are these numerous short oar-like structures that cover the surface of certain cells. They act like tiny paddles to help propel the cell or even just move surrounding fluid. Flagella, however, are much longer and much fewer in number, and they're primarily used to propel the entire cell forward, kind of like an outboard motor. To really understand how they move, we have to recognize the cell's internal scaffolding, the cytoskeleton. If you slice cilia or flagella open and look closely at their internal eukaryotic structure, you'll see a core called an axoneme. This core relies on that cytoskeleton, specifically these beautifully arranged parallel microtubules, to give them their structure and movement. Finally, we reach City Hall, Section 6, the nucleus and chromosomes. The nucleus, which was discovered by Robert Brown, is the ultimate heavily guarded control center of the advanced eukaryotic cell. It is deeply protected by a double membrane envelope, but its real importance lies inside. The nucleus houses the fundamental blueprint of life, the DNA. Our genetic material is stored here, and during cell division, this massive network of DNA tightly organizes itself into distinct, structured bodies called chromosomes. And here is a detail examiners absolutely love to test. Depending on where the centromere pinches them, these chromosomes take on specific types. They can be metacentric, submetacentric, acrocentric, or telocentric. This whole highly organized setup ensures that precise genetic information is passed on securely to the next generation of cells. It's the ultimate biological library. We've covered a massive amount of ground today, right? We moved from basic historical components all the way to highly complex cellular systems. But I want to leave you with a bit of a provocative thought. If complex life is entirely dependent on the highly specialized, perfectly coordinated city of a eukaryotic cell, how did the poor bacteria manage to survive and thrive with so little for billions of years? It really reinforces the sheer evolutionary brilliance of both simple and complex cellular units. Keep that curiosity alive.